Hey, in this video we're going to talk specifically about stem cells and how stem cells become differentiated. What are the processes in place that are required in order to turn on specific genes and to turn off specific genes. All of those discussions are going to happen, but we're going to talk specifically about cell specialization, cell differentiation, and application of stem cells. But we're also going to end the video with a kind of debate about the therapeutic use of stem cells, whether you're for it, whether you're against it. But I'm not going to try to persuade you. I mean, obviously you feel the way you feel, or you have opinions that are based on the way you feel. I'm just going to kind of highlight what the general for argument is and what the general against argument is as we go into this debate of stem cells. So let's dive in. Let's get started. Before we understand, or before we talk about stem cell use and stem cell differentiation, we need to talk about this idea of emergent properties. Emergent properties are all about properties that emerge as the result of the sum totality of the individual components. And what I mean by that is the whole is greater than any single component, or the whole organism is more complex. The whole organism can perform more complex functioning as the result of being produced from a variety of individual components. When you look at this diagram down here, obviously the organism is the sum total of the organ systems, which is the sum total of all organs, which is the sum total of all tissues, which is the sum total of all cells. Now, in previous videos, we've talked about kind of this hierarchy of organization. We talked about how cells within the cell theory make up the basic unit of life, and so that's why we start at step 1 with cells. Cells in this diagram are the least complex. They are the most basic unit of life, and so cells come together in similar or with similar cells. So for instance, heart cells, okay, the cells that make up the heart organ, the cardiac cells are going to come together with other cardiac cells to make cardiac tissue. The cardiac tissue is going to come together with other specific forms of cardiac tissue to make the entire organ that is the heart. The heart is going to work in concert with the other components, the other organs associated with the vascular or cardiovascular system, and that cardiovascular system is going to work in concert with the respiratory system. Those are going to work in concert with the endocrine system. They're going to work with the reproductive system. They're going to work with the muscular system, the digestive system, and when you put all of those systems together, you get the organism. Okay? When you put cells together with similar cells, you produce a tissue which is capable of producing functions that the individual cell cannot do. And as you go further into this complexity, each of these steps can complete a more intense, more complex functioning than the previous step, but it can only do those complex steps because they are made of individual components of the previous step. Does that make sense? So that is the emergent properties. An individual organism, again, I've said it numerous times, but an individual organism can only produce the functioning, or can only do the functions that an organism can do as the result of being produced from preceding organizational levels. Okay, so emergent properties arise from the interaction of the component parts, and the idea of this emergent properties is just kind of, to sum it up, the idea that the whole organism is greater than the sum of its parts. Okay, the whole organism is capable of doing more functioning than the individual parts that make it up. Okay, multicellular organisms are capable of completing functions that individual cells could not do, but is only possible because of the cellular functions of the individual cells. The organism can produce functions, okay, can do functions only because the individual organ cells that make it up. So, if we expand this a little bit, we obviously know that the cell is the basic unit of life, okay? So I'll put basic unit of life. And when you put multiple cells together, they make tissue. So if this is kind of step 1, this would be step 2, this would be step 3. step 4, and step 5. But if we were to expand this, okay, because we're looking at the kind of the holistic approach of biology, what are things that make up cells? Because cells are the basic unit of life, but they are made of subcellular stuff. When you go one level below cell, and for instance, if we expand it this way, what are the things that make up a cell? Well, you would say individual molecules, okay, and compounds. Those molecules and compounds, those organic molecules make up organic molecules. Those organic molecules make up membranes. They make up proteins. They make up carbohydrates. They make up phospholipids. They make up organelles. All of these things make up the subcellular, the things within a cell, the structures within a cell. So cells are made of molecules and compounds. Molecules, if we were to go this way again, are made up of individual atoms. Atoms are made of subatomic particles. Examples of those subatomic particles would include protons, neutrons, and electrons. Those subatomic particles actually are composed of quarks and other things, but I think we'll stop there, right? So if we expand this all the way out, subatomic particles make up atoms. Atoms come together to make molecules. Molecules come together to make cells. Cells make tissues. Tissues make organs. Organs make organ systems. Organ systems make organisms. Organisms, if we were to expand it out this way, because this is just kind of a very broad, narrow spectrum of the entire hierarchy of organization. Organisms come together to make a population of those specific organisms. Populations come together to make communities. When you put individual populations of, let's say, people with populations of rabbits, with populations of dogs, with populations of deer, when you put the communities together with the abiotic or non-living factors within a particular area, it makes an ecosystem. And when you put multiple ecosystems together, you get a biome. And when you put multiple biomes together, you get the biosphere. Right? So this is the entire spectrum from here all the way through this kind of narrow band, all the way to Earth itself. So from the very level of subatomic particles like protons, neutrons, and electrons, all the way through atoms, molecules, cell, tissue, organ systems, organisms, and all the way through kind of this population and ecology section. This will be the focus when we get into the ecology in second year IB, but for now, we'll just go ahead and label this ecology, and we'll get there. But know that it's here at this point. So we've expanded this. This is kind of the section we're focusing on within this cellular biology topic, but for now, let's move on. Cell differentiation. So we've talked about the emergent properties. We've talked about kind of an intro into cell bio, but now we need to talk about how cells actually differentiate. And the way that cells are going to differentiate and specialize is through gene expression. This is a very, very small section of a genome within the cells. One thing you're going to want to know off the bat is that all somatic cells, okay, all of the diploid cells that make up an organism like you, originated from one cell, the zygote. So remember, one sperm cell comes together with one egg cell. This egg cell has 23 chromosomes. The sperm cell has 23 chromosomes. They come together during fertilization and produce a single cell that is 46 total chromosomes. That is the zygote. Okay, that's the zygote. From that single cell, it divided and produced two cells that each have 46 chromosomes. Those cells divided into two cells each. Okay, and eventually you get to an organism through the fetus, through the baby, through the adolescent, through the toddler stage that eventually has several trillion cells. Okay, but all of these several trillion cells that make up you through several rounds of cell division all originated from the same cell. And that cell is called the zygote. Now, because all of your individual body cells that originated from the single cell, or because they originated from a single cell, they're going to originate from the same original copy of DNA. So this DNA, okay, the genome within this cell that was the first cell that made up you, contains only one molecule of DNA. Well, that molecule of DNA is going to replicate itself through a process called DNA replication. It's going to give one copy to this cell. It's going to give one copy to this cell, and because subsequent cells through several rounds of cell division are going to continue to copy the same DNA, it's going to make one universal kind of copy of DNA that makes up you. So every cell in your body has the same genome. Now, because every cell in your body has the same copy of DNA, that means every cell type has the ability to become every cell type, right? So your skin cells, for instance, have the genes necessary to make those cells liver cells. And liver cells have the genes that are necessary for making nerve cells. And nerve cells have the genes that are necessary for making muscle cells, and so on. We have a lot of different cell types in our body. All of them have every gene responsible for making each other, which is why we're going to start or eventually get into this idea of stem cells, which have the ability to differentiate in some cases into any potential cell, because they do in fact have the instructions for any cell. But for now, know that the diploid cell, the original zygote, the original diploid cell gave rise to every cell in your body, and therefore all the DNA within every cell in your body is the same. So there's a common genome. How do we then differentiate if every cell has every set of instructions or the same set of instructions? How do you actually specify which genes are turned on and which genes are turned off? And so it's all about cell signaling. Cells kind of right after production, right after a cell is produced, right after a round of cell division, for instance, this cell, let's say, was a skin cell. It's going to divide and it's going to produce 2 skin cells. I mean, that's important. We don't want to produce skin cells in our liver, and we don't want to produce muscle cells in our neurons, When we need skin cells, we need to replicate and produce skin cells. So the original skin cell, when it divides into 2 new skin cells, is going to quickly send signals. Therefore, the new cells that were just produced are going to quickly receive those signals, and the signals are going to signal the deactivation of specific genes. It's that simple. We want to deactivate genes we don't need, and therefore leave active the genes we do need. So if we are deactivating unneeded genes and we are leaving the needed genes active, it's going to correlate to proper cell functioning. Deactivate genes that are unneeded, and leave active the genes that are needed. We're going to ensure that the cell will specify or differentiate into the correct cell type. So, why is this important? Nerve cells have to function completely different than skin cells. When you look here, nerve cells look very different from skin cells, and skin cells look very different from nerve cells. Both of those look very different than muscle cells. All 3 of these particular types of cells have specific functions that are required in order for that particular cell to function correctly, right? They have specific jobs. Epithelial cells are perfectly structured to allow the cells to work as a boundary between the inside of the body and the outside of the body. Nerve cells don't have to be a boundary between inside and outside of the body, so they don't structure themselves like a boundary. They structure themselves to be very long and drawn out because their job is to send chemical and neurological signals from one part of the body to the other part of the body, right? So they're all about neurotransmitter release. They're all about neurotransmitter uptake. They don't function like epithelial cells and therefore aren't structured like epithelial cells. Muscle cells are all about contracting and relaxing. Muscle cells are all about moving the organism from one place to another. They don't need to uptake or worry about the uptake of neurotransmitter. They don't need to worry about creating a boundary between the inside and outside of the body like epithelial cells do. So 3 completely different specialized cells that are perfectly structured to do the job that they are required to do. One of the things that epithelial cells produce and maintain is this pigment molecule known as melanin. Melanin is something that is specific to epithelial cells because it is one of the ways that we help protect our tissues from the damaging effects of UV radiation. Again, because they are on the outside of our body, they produce the melanin. We don't need melanin within interior tissue cells. And so the nerve cells and muscle cells, for instance, turn off the gene necessary to produce melanin and therefore don't produce it. The epithelial cells keep the gene on that allows them to produce melanin because, again, that's one of the things that they need to structure themselves with, and so they have the ability to keep that gene active and therefore produce it. Epithelial cells do not require the ability to release and absorb neurotransmitter molecules. We did that as well. or we talked about that already. Epithelial cells will turn that gene off specifically and therefore deactivate the gene. Okay, so when you look at this quick kind of individual small section of this undifferentiated cell's genome, this is a section that contains 6 genes. The X's indicate unexpressed genes, so these genes specifically would be turned off. The number of arrows would be the degree by which the expression happened, right? So 3 arrows is going to be highly or more highly expressed than 1 arrow, for instance, but gene A and gene C, for instance, in this particular diagram would be expressed. And so you can see that it's just a different code, right? All 3 of these specialized cells have the same 6 gene sequence, but when, for instance, we turn or when we deactivate gene B, C, and E and keep A, D, and F, when we keep these particular genes active and keep the degree of expression this way, this cell specializes and differentiates into an epithelial cells. If you were to compare that to the nerve cell with gene A, B, C, D, E, and F, if we keep E, F, A, and B on and deactivate C and D, this is a different sequence than this. It's a different deactivation and activation sequence, and so the nerve cells produce different structures and therefore function differently. And then to compare both of those with the muscle cells, you can see that the muscle cell activated or kept active the A gene, D, E, okay, D, E, A, and deactivated B, C, F. So 3 different cells, 3 different specialized cells, 3 different special structures, 3 different specific functions. All of them deactivated certain genes within the same sequence, which gave rise to the specialization and the differentiation that you can see, which leads into the talk about how specialized an organism is. We as humans, and which is really not uncommon for a lot of other organisms, have 220 distinct and highly specialized cell types that have currently been recognized. When you look through the human body, there are 220 or have been 220 classified specific special cell types, all of which have the exact same set of DNA, okay, because they originated from the same diploid zygote. And all 220 of these highly specialized cell types have highly specialized cell structures. Each of these cells, as you can see in this diagram, which only looks at a few, have specific structures that allow them to function correctly. All of them have the same set of genes, but they all turn off a specific sequence or a specific number of genes to therefore leave active only the genes that are required to function correctly. So all specialized cells are produced from the activation and deactivation of specific genes, leading to unique combinations of activated genes for all specific cell types. Muscle cells turn off the genes that are specific to being a blood cell, and blood cell turns off the genes that are specific to being a muscle cell. Blood cells also turn off the genes specific to being nerve cell, and nerve cells turn off specific genes for being liver cells. Okay, and so you can say that same thing for all of these cell types. 220 distinctly highly specialized specific cells that all differentiate and specialize from a common genome by turning off specific genes and leaving active certain genes. Specialized cells have unique structures that allow them to carry out specific and very specialized functions. The more specific a specific cell, okay, or the more specified the specific cell's function, the more deactive genes, or the more genes that were deactivated to make that cell more specific. Okay, so let's get into what a stem cell is, the different levels of stem cells, what stem cells can eventually become, and then we're going to get into the uses of the stem cells as well. So, what does it mean to be a stem cell? Stem cells are cells that have not yet been differentiated or specialized. And so stem cells are undifferentiated and unspecialized. Really important. Undifferentiated, unspecialized. And because they're undifferentiated and unspecialized, they have the ability to become a variety of specialized cells, right? You're basically taking the raw product, okay, the raw reactant, the raw good, and you're refining it through a series of reactions to produce a particular special product, okay, finished product that has a specific job and a specific structure that is well suited to do that specific function. So all of our lung tissue cells, our pancreas tissue cells, our muscle cells, our red blood cells, our skin cells, our neurons, our brain cells, and so so forth, are all produced through specialization, but they're all produced the first time through or from the embryonic stem cells. Okay, so what are the different varieties of stem cells? We have totipotent, pluripotent, multipotent, unipotent. What do they mean? Totipotent can differentiate into any cell type. This is a totipotent stem cell. Okay, embryonic stem cells are totipotent stem cells. They have the ability to produce any cell type, which is why it's on top. This one cell can differentiate into any of the specialized cells that eventually become an organism. Pluripotent is the next level. They can differentiate into many cell types. Okay, plura, remember, is many. Tota is any. So totipotent can produce any cell type. Pluripotent can produce many cell types. Multipotent can differentiate into only a few cell types, but the few cell types have to be kind of closely related to each other. And then unipotent can only differentiate into one specific cell type. So what does it mean? We can look at this graph over here, or diagram, in order to kind of understand more about what these stem cells can do and what each of the levels of organization, so to speak, mean. When we look at totipotent embryonic stem cells, notice that they have the ability to differentiate into anything. Next line would be the pluripotent. They have the ability to specialize into a variety, okay, many different varieties within each of the lines, but notice that not all cell types can be produced from each of the pluripotent embryonic stem lines. We have ectoderm line, mesoderm line, endoderm line. These are the three kind of tissue locations or tissue types within an embryo that is developing. The endoderm line is a pluripotent embryonic stem cell. It has the ability to produce many different cell types, but all of these cell types will eventually become a variety of cell types that only the ectoderm line can produce, right? And so the mesoderm line, pluripotent embryonic stem cells can only then produce the specific cell types that are specific to the mesoderm line. And the endoderm line can produce the variety or the cell types that are unique to this endoderm line. So while the totipotent embryonic stem cell can produce all varieties of cells, the pluripotent embryonic stem cells can produce just the variety of cells within each of those lines. And then multipotent stem cell, dropping down one more level, can produce a few each, but remember, the few that they can produce are very closely related types of cells to each other. And so we've got multipotent stem cells at this level, and then you have the actual differentiated or specialized cells at this level. So totipotent, pluripotent, multipotent, and unipotent. These unipotent stem cells are still undifferentiated, so they're still stem cells, but they only have the ability to differentiate and specialize into one unique cell type. So we've talked about what stem cells are. We've talked about kind of why they're needed within embryonic development. Now we need to talk about how we could potentially utilize them to help within kind of a medical context, right? So the two disorders that I would like to talk about are Stargardts Macular Dystrophy and leukemia. Stargardts Macular Dystrophy is a problem associated with the retina within the eyeball, and leukemia is a form of cancer specific to the blood system. And so if we dive into what this Stargardts Macular Dystrophy is, it's a recessive genetic disorder. Okay, so it comes about as the result of the assortment of two recessive alleles. It affects 1 in 10,000 children, and it's just a rare recessive mutation that causes one or multiple channel protein pumps that are on or associated with the membrane of the retina to malfunction. and because those membrane protein pumps can't work correctly, they can't properly transport energy that is required in order for the photoreceptors to work properly. And so if they're not working properly, it leads to a progressive and eventually a totality in terms of the loss of central vision. So you can see it in here. There is kind of a degenerative eye disorder associated with the retina. And you typically, when looking at somebody's retina, can diagnose or determine that Stargardts macular dystrophy is in fact the case, because there is often a place or kind of a central location on the retina that is essentially dying. Now, how can stem cell treatment work or impact the patient's outcome? It's all about employing embryonic stem cells or treating these embryonic stem cells to differentiate specifically into retinal cells before just injecting or replacing the patient's bad retina cells with those new and improved retina cells. Once those new retina cells that came about or that were produced from the embryonic stem cells or differentiated or specialized from the embryonic stem cells are injected into the patient's retina, they quickly kind of adhere or attach themselves to the retina and start functioning correctly, or at least that's the goal. So by employing or treating these embryonic stem cells and then injecting them to replace the bad or damaged retina cells, the improperly working retina cells, we hopefully can try to get the new and improved stem cells to start functioning correctly, and therefore leading to an improved central vision for the patient. So, good use of stem cells in my opinion. As we go into leukemia, leukemia is a disorder that is affecting a patient's blood or bone marrow. The leukemia is a cancer specifically of the blood. So as a result of the cancer, the white blood cells are really ramped up because we know that white blood cells are associated with the immune response of a particular person. And so if there is a disorder that is detected by the person, it oftentimes is accompanied by increasing the concentration of these white blood cells. However, due to leukemia, these white blood cells typically have poor functioning within themselves, and so they really don't work correctly, and so it's not an effective treatment or not an effective way of handling this really invasive disorder. So how can stem cells treat or help in the treatment of leukemia patients? Stem cells for leukemia specifically are harvested from the patient's bone marrow. And again, we're talking bone marrow of the patient. So we're going to take stem cells harvested from the bone marrow of the leukemia patient. Those can come from peripheral blood as well, or they could come from umbilical cord blood from babies that parents have allowed to donate umbilical cord blood. All of these sources contain stem cells. The goal of this stem cell treatment is basically to get stem cells, then subject the person to chemotherapy and radiation so that they can destroy the diseased white blood cells, right? These destroyed white blood cells need to be replaced with healthy white blood cells because the chemo and the radiation has substantially dropped or decreased the concentration of these white blood cells. And people naturally need a minimum concentration of these blood cells in order to properly function. Once we kind of knock out a large portion of those damaged or diseased white blood cells, we're going to then take the harvested stem cells, inject them back into the patient's bone marrow, allow them to differentiate into new healthy white blood cells, and therefore hopefully will just kind of replace and fix the bad, often mutated white blood cells that have led to leukemia, and in the hopes of completely curing, ideally, the person from this disease in the first place. So it is low risk of immune system rejection due to using those patient's own stem cells. I mean, if we are harvesting stem cells from the patient, keeping them kind of held aside, knocking all of the white blood cells down or knocking out all of the diseased white blood cells, and then putting the patient's own stem cells back into the bone marrow and allowing them to differentiate and become properly functioning white blood cells, is the whole goal and the whole treatment protocol at this point, to try to eliminate this disease from a person's body and therefore curing them permanently. Another kind of cool use of stem cells in kind of medical research right now. So Last thing we need to talk about is kind of the ethics behind stem cells and kind of the pros and cons for utilizing them in not only research but medical applications. So there are 3 stem cell sources really that I would like to talk about. One of them is embryonic stem cells, one is cord blood stem cells, and the other one is adult stem cells. So there's a variety of descriptors over here on the left. We're going to kind of just talk through these descriptors within each of the stem cell sources. Starting with ease of extraction. So this is how easily the stem cells are extracted from that particular source. When we talk embryonic stem cells, they only can be harvested from embryos. I mean, that's what embryonic stem cells mean. So you can't get embryo stem cells from toddlers. You can't get embryo stem cells from adults. You can't get them from children. You can't get them from donors. You have to get them from embryos. And we're talking early embryonic development, before a lot of the differentiation and specialization has happened. So they can only be obtained from embryos, and most of the time they're generated from embryos that were generated in excess in a lab via the in vitro fertilization programs. So those programs, which we'll talk about later in the syllabus, are all about trying to make a variety or a high number of viable embryos in a lab so that one or a few can be impregnated or at least implanted back into the female's uterus in order for it to eventually grow and allow the parents or the couple to give birth to a normal healthy baby. So the ease of extraction at this point is, what do you do with all of these excess embryos? Well, one of the things that perhaps could be a positive out of the excess embryos generated by these programs could be to use them as a source of stem cell, embryonic stem cells. Cord blood is very easily obtained. It is very easily stored, but it is very limited in the quantities available. 1, because parents have to give consent, but 2, there isn't much blood within the cord within a baby that is produced. So easily obtained, easily stored, but again requires consent. Adult stem cells are very difficult to extract just due to how deep into the tissue they really are. I mean, we're talking about really getting into the bone marrow and the deepest parts of the tissue with a needle in order to extract these stem cells, which are often also in very low quantities, just because an adult doesn't really require regenerating a lot of these stem cells. So they're very low quantities and they're available only within really deep tissues like bone marrow. Ethics of the extraction. So we've already talked about how easily they are extracted from the specific sources, but the ethics associated with the extraction is really important. When we talk embryonic stem cells, they really can only be obtained from or by destroying the embryo. So you cannot, at this point in our medical research or our medical practice, you cannot extract embryonic stem cells and maintain life of the embryo. So you cannot keep the embryo alive, therefore have to destroy the embryo in order to extract those stem cells. So the big ethics here is the embryo can't give consent on its behalf. So if the embryo, which will eventually give rise to a normal human, right, if it can't give consent, at what point is consent kind of given to the researchers, is it given to the scientists, is it given to the parents, that kind of thing. Again, ethics. Cord blood, the consent is required and is required of the parent. So parent consent is required to harvest stem cells. The umbilical cord is removed at birth and discarded whether or not stem cells are harvested. So this is a normal part of childbirth. The cord is just discarded. Parent consent is required, but is a viable source of stem cells. And so I guess the ethics associated with this is, if this material is going to be removed and discarded at birth, should it be employed to potentially save others from diseases like leukemia? I don't know. Something for you to decide. The adult patient obviously can give consent, and so really no ethics associated with adult stem cells. Adult patient can give consent. Yes or no, I want you to harvest my own stem cells. I want you to put the stem cells back in me, blah, blah, blah. You can give consent. Really no ethics there. The growth potential of embryonic stem cells is almost unlimited because the embryos haven't had an ability to or time to differentiate or specialize at this point. They can almost unlimited grow or have unlimited growth potential. The cord blood and adult, because they're a little more specialized at the time of harvest, have much reduced growth potential, but they still are stem cells and have the ability to grow at a lower risk of developing a tumor. Because these are almost unlimited growth potential, they have a higher risk of tumor development. Remember, tumor is essentially just unregulated cell growth. So if you have an unlimited growth potential, you naturally are going to have a higher risk of tumor development. If you have reduced growth potential, you're just naturally going to have a lower risk of tumor development. The differentiation that can happen between embryonic cord blood and adult stem cells is that embryonic stem cells can differentiate into any cell type. Remember, the embryonic stem cells are those totipotent stem cells, so they can differentiate into any cell type. The cord blood, again, like I said up here, have reduced growth potential. They are a little more specialized or a little more differentiated at the time of harvest. They have limited capacity to differentiate, and it requires oftentimes the inducement due to cells naturally wanting to divide into blood cells. So the stem cells that come from the cord blood are programmed naturally to become new blood cells. So we have to take those kind of normally differentiated blood cells, or what would eventually become normally differentiated blood cells, and we have to, through gene therapy, kind of turn on the genes we want to turn on and turn off the genes we want to turn off in order to kind of reprogram these eventual blood cells into other forms of cells that we could use for a variety of other reasons. Adult stem cells are much more limited to differentiate or have a much more limited capacity to differentiate, and blood or adult stem cells very much are dependent on source tissue. So oftentimes if you're going to get cells out of the bone marrow, they're going to eventually become blood cells because that's ultimately what the bone marrow was going to differentiate into. Now when we get into things like genetic damage, obviously there's going to be more accumulation or a longer time for these mutations to accumulate through the life of an adult as the age of the adult increases. So we know that mutations happen for a variety of reasons, right? Random mutations happen all the time. Exposure to UV radiation can cause mutations. Exposure to viral infections can cause mutations. Carcinogens in the environment can obviously cause mutations as well. And so as an adult lives through its lifetime or through its lifespan, the longer the adult is alive, the more likely genetic damage will occur. And so the age of the adult, or the age of, therefore, the stem cells, or the age of the cells that you're taking, age of the tissue that is made of the stem cells that you're harvesting, oftentimes have an accumulation of these mutations which will eventually manifest themselves as expressed traits once they differentiate and specialize. Now, because embryos are very young, there's much less chance of genetic damage than there is with the adult stem cells, just based purely on age. The compatibility of the sources of stem cells is different as well. Stem cells are not genetically identical to the patient because we are taking them from an embryo that obviously isn't the patient. So there isn't any kind of genetic compatibility. However, when cord blood or adult stem cells are harvested, they can and often are genetically identical to the patient because, like I said in the leukemia discussion, you can actually, as an adult or a child, harvest your own stem cells, hold them aside, go through rounds of chemo and radiation, and then just inject those same stem cells once they specialize and differentiate back into the patient. So they are, in fact, genetically identical to the patient. Now, very last thing we're going to do to kind of finish off this entire kind of ethics discussion is talk about the arguments for and the arguments against therapeutic cloning. Now, therapeutic cloning is slightly different than stem cells, but it definitely incorporates and utilizes stem cell at the foundation of what this therapeutic cloning is all about. So, if we run through the arguments for therapeutic cloning, again, these are the arguments for. I put them in green. This is kind of like pro-therapeutic cloning. This is red. It's kind of like against therapeutic cloning. Arguments for therapeutic cloning is that stem cell research may lead to beneficial future discoveries, right? Stem cells have already been a part of the treatments for not only Stargardts Macular Dystrophy, but also leukemia. We have, over the course of the last 10 years, really made huge strides in childhood cancer research. We've also made huge strides in just disorder, random disorder treatments as the result of trials that are associated with stem cell research. So it is likely that the stem cell research may lead, in fact, to beneficial future technologies that we don't even know exist yet. They may be used to cure serious diseases or disabilities like cancer. Okay? Transplants are less likely to be rejected as they are genetically identical to the parent. This therapeutic cloning program is all about harvesting stem cells essentially from the patient, holding them aside, allowing them to differentiate and specialize into specific tissues and specific components of that particular patient, and then using them potentially as like whole organ transplants for that particular patient. Transplants do not require the death of another human. Typically right now, in order for a human to get a liver, for instance, or to get a heart, for instance, or lungs, for instance, they have to actually be taken out of a deceased donor. Whereas if we could just utilize stem cells from the patient, grow an additional organ outside of the body, we could essentially transplant that organ into the person as its own growing tissue that is genetically identical to the tissue that was already there, and it doesn't require a wait list, and it doesn't require death of another human. Stem cells can be taken from embryos that have stopped developing and would have died anyway. So this is talking, one of the reasons that are pro stem cell research is that these embryos would have died and are going to continually die due to abortion, due to in vitro fertilization programs. And so the argument is, if the stem cells or the embryos, for instance, are going to die anyways, why not harvest the stem cells from them and use them in a positive way? Okay. Cells are taken at a stage when the embryo has no nervous system and can arguably feel no pain. So if the embryo is going to die and the embryo at that age doesn't feel any pain and doesn't have an intact nervous system, again, even more reasons within this argument to go ahead and be pro stem cell research. Stem cells can be created without the need for fertilization and destruction of natural human embryos. That gets kind of away from the killing another human mentality, right? By saying we can create embryos by artificial fertilization methods, and we therefore can go against, or we can go against the need, okay, of destroying these naturally formed human embryos, and it's called induced pluripotent stem cells. Basically, induced means that we're artificially going to create them, and therefore don't have to destroy naturally produced human embryos. Arguments against is all about kind of the opposite of what was said in this green section. So arguments against therapeutic cloning is all about, it involves the creation and destruction of human embryos. So again, we are killing life. And so at what point do we afford the right to life? Obviously, the embryos can't give consent. Oftentimes, parents don't or can't give consent at this point because the embryos were created outside of the lab or outside of the body within a lab situation. Embryonic stem cells are capable of continued division and may develop into cancerous cells and cause tumors. Again, that's an against. That is a negative, is that because there is increased risk of tumor development, we may in fact be putting cells into a person that will eventually give that person cancer. More embryos are generally produced than are needed, so excess embryos are killed. By going to or towards this therapeutic cloning program, we are essentially going to ramp up the number of embryos that are developed, and therefore we're just naturally going to ramp up the number of embryos that are killed. And so the argument against therapeutic cloning is, at what limit are we going to set, or at what point do we afford the right to life? Like, if we're going to continually ramp up the number of embryos developed and therefore going to continually up the number of embryos killed, it's just kind of like this downward spiral on putting a de-emphasis on life in general. With what additional cost and effort, alternative technologies may fulfill similar roles. So nuclear reprogramming of differentiated cell lines. This is all about instead of producing an embryo to kill it, this is an additional alternative technology that is really trying to fulfill the same role, and it's all about reprogramming differentiated cell lines. So lines of cells that have already differentiated and specialized, but going into the nucleus and reprogramming them to become something they're not. So if we can put all of this time and effort and cost into learning how to reprogram cells, maybe we don't need to go down a path of producing embryos to therefore kill them, if all that really needs to happen is just to reprogram the computers, so to speak, that are already there. Then, of course, as an argument against, we get into the religious and the moral objections due to the playing God argument. Okay, there are a lot of people that have really staunch or stout religious beliefs that life is really created and destroyed by God, and therefore needs to be left to that. And so by creating embryos and killing embryos, you're essentially playing God, and therefore science shouldn't go there. The embryo which is created could potentially be used in vitro fertilization and develop into a human fetus. So the question is, are we creating human life to destroy it? I don't know. That's up for you to decide. And although cloning humans reproductively is illegal, currently, this has not been ratified by all nations, and so you get a potential for a race to clone the first human. There are obviously going to be some countries that are pro-cloning. There are going to be some countries that are against cloning. And so if we kind of opened up this therapeutic cloning as kind of mainstream, it might potentially give rise to the race to clone the first human, and that could have potentially serious repercussions for the class of organisms that are called humans. So just some things to think about, some arguments that are for therapeutic cloning, some arguments that are against the therapeutic cloning. Hopefully I didn't persuade you. Hopefully you're keeping an open mind. Hopefully you at least are a little more educated about what therapeutic cloning is, what stem cells are, when stem cells can be helpful, what the sources of these stem cells are, what the levels of stem cell development are, and stuff like that. So bring your questions to class. See ya.